Photosynthesis is one of the most important metabolic processes on Earth. We're going to begin with an overview of the process of photosynthesis, beginning with a summary equation. A summary equation is a way of boiling down a metabolic pathway into a simple expression showing the reactants and products. Here we see carbon dioxide and water are the reactants. With the presence of light energy, glucose and oxygen are the products. The previous slide was an unbalanced summary equation. This one is chemically balanced, indicating we need 6 CO2s, 6 water molecules to produce 1 glucose and 6 oxygens. Notice also that the process involves both reduction and oxidation stages. Carbon dioxide is reduced, water is oxidized. You, my friends, are heterotrophic because you must consume your food. Autotrophic organisms are those that can take inorganic molecules and create organic molecules. In an ecological context, we call photosynthetic creatures producers because they produce the food consumed by all the rest of the creatures. Producers are found in 3 major taxonomic kingdoms that would include the bacteria or prokaryotes generally, in algae, and in plants. On land, plants are the primary producers, but in aquatic ecosystems, it's photosynthetic bacteria and algae that are doing most of the production. Remember our origin story for chloroplasts. Chloroplasts arose through endosymbiotic relationships between ancient eukaryotes and photosynthetic bacteria. So plants and algae can only conduct photosynthesis in the tissues that contain chloroplasts. Chloroplasts themselves have anatomy. They have a region called the stroma, a fluid-filled space where carbon dioxide is reduced, where it is fixed. Remember, the idea of fixing a nutrient is converting it from a less useful to a more useful form. Reduction is the addition of electrons and hydrogen ions. So this is the place where carbon dioxide becomes a carbohydrate. Thylakoids are the membranes within the chloroplast that contain the pigments that drive photosynthesis. That is chlorophyll. Here's a figure you might remember looking at in lab when we looked at a cross section through a plant leaf. You can see within the leaf there is the photosynthetic tissue called the mesophyll. The bottom image is a cartoony interpretation of a chloroplast, and then finally an electron micrograph of a chloroplast. Animals are heterotrophs, right? Well, there's always an exception. This beast is a photosynthetic slug, a mollusk. It actually consumes algae, takes the chloroplasts from the algae cells, transports them to its own tissues, and becomes photosynthetic. Who doesn't like a rainbow? Rainbows are natural examples of the fact that white light is made of a spectrum of colors. Here we see the spectrum of energy coming at us from the sun. Visible light, or white light, is made up of a spectrum from violet to red. It's sandwiched in between ultraviolet, which is wavelengths that are shorter and higher energy than white light, and infrared, which is energy made up of longer wavelengths and lower energy than white light. Photosynthesis is driven by solar energy. Photons of light are particles that travel as waves, and when these particles strike pigments in the chloroplasts of plants and algae, those pigments get excited, and the excitation of those pigments and the electrons they release are what will fuel photosynthesis. This image depicts a chloroplast being subjected to light energy. Plants are green because chloroplasts are green. Chloroplasts are green because chlorophyll is green. Chlorophyll is green because it reflects or transmits the green part of the spectrum. The violets, blues, reds, and yellows are absorbed. Those are the parts of the light spectrum that actually drive photosynthetic activity. So it's a little counterintuitive that green is the least useful part of the white light spectrum when it comes to making photosynthesis happen. I want you guys to think about our photosynthesis lab, where we extracted pigments from spinach leaves and used chromatography to separate them. Check out frame A on this graph. It shows absorption of different wavelengths by different kinds of pigments in plants. So we see chlorophyll A most efficiently absorbs purple, B absorbs blue along with the carotenoids, and to a lesser extent the reds and yellows. So purples and blues, reds and yellows, that's what makes photosynthesis happen. Frame B shows the amount of oxygen released if plants are exposed to different spectra of light. So lights that emit more purples would make more photosynthesis happen and make more oxygen. Frame C was a pretty famous experiment done by a botanist long before we understood the details of photosynthesis, where he took a string of algae, put it on a microscope slide, added some aerobic bacteria, oxygen-loving bacteria, then shone a light through a prism and let that spectrum fall on the microscope slide. And what he saw is the bacteria migrated to the purple end and the red end, where most of the oxygen was being produced. Within the thylakoids of a chloroplast, chlorophyll molecules are organized into photosystems. So a photosystem is a whole complex of chlorophyll molecules. This complex absorbs photons of light, channels those photons to a centrally located chlorophyll molecule that gets so excited that it starts to emit high energy electrons, and these electrons are passed off to an electron transport chain. So here's a close-up of a photosystem. So notice the phospholipid bilayer that makes up the thylakoid membrane, the complex of chlorophyll molecules. The arrow indicates how an energy-rich electron is handed off to an electron transport chain, and just like in aerobic respiration, the electron transport chain is used to make ATP, and we will see that that ATP helps drive the reduction of CO2 into a carbohydrate later in the pathway. So in spite of the fact that we can express photosynthesis as a summary equation, remember it's really a long metabolic pathway, a series of linked reactions. This illustration shows us that the pathway plays out in two major stages. On the left side of the image, we have the involvement of thylakoids and light and water. That is what I refer to as the light-dependent reactions. On the right side, we have the pathway by which CO2 is fixed. I refer to that as the light-independent part of the pathway. And notice the two are linked. The products of one become the reactants of the other, and vice versa. Our learning objectives for this unit include being able to characterize what's happening during the two parts of the photosynthetic pathway. Where does it occur? What are the reactants? What are the products? You might find it useful to build for yourself a little input-output table for both the light-dependent and the light-independent reactions. So these light-dependent reactions are happening inside the thylakoids. They involve chlorophyll molecules getting excited as photons of light strike them, energized electrons being released, passed off to an electron transport chain that produces ATP and reduces the coenzyme NADP to NADPH. So NADP is an important coenzyme in photosynthesis that plays the same role as NAD does in cell respiration. The light-dependent reactions also require water. Water provides a source of hydrogen ions and electrons, and when you split water up, oxygen is released. So the oxygen that is a product of photosynthesis is traced back to these light-dependent reactions. So as chlorophyll molecules are getting excited and losing electrons, water serves as a replacement for those lost electrons. So here's a simple visual that would be appropriate for constructing an input-output table to summarize what's happening in the light-dependent reactions. So notice the inputs include light, water, NADP, and ADP. The outputs include oxygen, NADPH, and ATP. Of those three outputs, the latter two will be inputs to the light-independent part of the pathway. So here's a more detailed look at the light-dependent reactions. Notice there are two photosystems involved, called photosystem 1 and 2, and light is striking both simultaneously. Both are getting excited and losing electrons. Photosystem 2 replaces lost electrons by splitting water. Its electrons are passed off to the electron transport chain to make ATP. Photosystem 1 accepts the used electrons at the end of the electron transport chain to replace its lost electrons. Photosystem 1 hands electrons off to the coenzyme NADP. Here's a cartoony interpretation of the way the two photosystems are linked by way of electron transport chains. So we can see how photons of light striking the photosystem produces the energized electrons, how photosystem 2 passes off electrons to the electron transport chain, and how photosystem 1 hands off electrons to NADP. Both the NADP and the ATP will be used as reactants in the second part of the pathway called the light-independent reactions. There are many interesting similarities between chloroplasts and mitochondria, between photosynthesis and aerobic respiration. One of them is the use of electron transport chains and ATP synthase. In this figure, we see the portions of the organelles where hydrogen ions are pumped to during the electron transport chain activity. Those are the areas shaded dark, and the areas to which hydrogen ions diffuse during ATP synthesis. That's the light area. So it would be the matrix of the mitochondria and the stroma of the chloroplast. One more look at the details of the light-dependent reactions here. Please note that there are many photosystems and many electron transport chains scattered throughout the thylakoids within a chloroplast. Also, pay attention to how the NADPH and the ATP made are going to be inputs to the Calvin cycle, which is the sequence of reactions that make up the light-independent part of the pathway. All right, now on to the light-independent reactions. These reactions are happening inside the fluid space that surrounds the thylakoids called the stroma. In these reactions, the ATP and the NADPH is used to reduce carbon dioxide. That process requires energy supplied by ATP. It requires hydrogens supplied by NADPH. Carbon dioxide will be reduced into a carbohydrate. This is a cyclic pathway. So recall in aerobic respiration, we had the citric acid cycle. Here, this cyclic pathway is called the Calvin cycle. So this figure is sufficient to create an input-output table to summarize the light-independent reactions of photosynthesis. The inputs include carbon dioxide, ATP, and NADPH. Carbon dioxide provides the carbon and oxygen. ATP provides the energy. NADPH provides the hydrogens. The outputs include sugar. In our summary equation, that would be glucose. It would take 6 CO2s to make 1 glucose. But we also have ADP and NADP as outputs. And if you follow the arrows, you will see that those are inputs to the light-dependent reactions. Cells have a limited supply of these coenzymes, and they are used and reused over and over. The slide gives us a more detailed look at the actual sequence of reactions that make up the Calvin cycle. The process begins with the input of 3 carbon dioxide molecules. An enzyme called Rubisco will catalyze the combination of those carbon dioxides and 3 molecules of a 5-carbon phosphate sugar called RUBP, ribulose biphosphate. So there's a phosphate group on both the number 1 and the number 5 carbon. The product of that is 6 molecules of a 3-carbon phosphate sugar called 3-phosphoglycerate. Some of the ATP made in the light-dependent reactions are now used. Those phosphate groups are transferred to the 3-phosphoglycerate, converting that into 1,3-biphosphoglycerate. Some of the hydrogens that were produced in the light-dependent reactions are used. NADPH will transfer those hydrogens, and the 6 molecules of 1,3-biphosphoglycerate become 6 molecules of G3P, glyceraldehyde 3-phosphate. The cell gets 1 of those as the net gain. Remember, this is a cycle, so the other 5 molecules must remain behind to allow the cycle to continue. Those 5 molecules of G3P are phosphorylated using more of the ATP made in the light-dependent reactions, and those 5 G3Ps are converted into the 3 molecules of RUBP we started with. So now technically, the output of the Calvin cycle is actually 1 molecule of a 3-carbon sugar called G3P. 2 turns of the cycle, a total of 6 CO2s would be required to fix enough carbon to put those 2 G3Ps together to make 1 glucose. So we saw this figure both in lab and earlier in this presentation. Focus on the lower epidermis of the leaf, and we see these openings called stomata. Stomata allow gas exchange to happen. Plants have the opposite gas exchange need we have. They produce a net amount of oxygen, which they must get rid of, and they use a net amount of carbon dioxide, which they must gain. So again, those stomata provide openings through which CO2 enters and O2 escapes. In the previous slide, we saw that when that carbon dioxide enters and is used in the Calvin cycle, the very first product that we can detect is a 3-carbon molecule. So for plants like that, we refer to them as C3 plants. The plant depicted in that previous slide was a cottonwood tree. Cottonwood trees are C3 plants. Like all plants, as long as their stomata are open to allow gas exchange to happen, they also lose water. It's called transpiration. Now, C3 plants run into trouble when the weather or the climate gets too hot and dry. As a way to prevent excess water loss, they close their stomata, but that shuts off the supply of carbon dioxide. So there are plants that thrive in hot, dry climates. One group of these are called C4 plants, and a commercially important example of a C4 plant is sugarcane. C4 plants build up a reservoir of carbon by fixing CO2 prior to the Calvin cycle, creating a 4-carbon organic compound that can later be broken down into CO2 and provide the carbon that will allow the Calvin cycle to continue even if they have to close their stomata. So they can close their stomata when it's hot and dry and still have this prefabbed reservoir of carbon in the form of these 4-carbon compounds. A second adaptation is seen in plants called cam plants. These tend to be thick, succulent plants like cacti and pineapples. Cam plants have the strategy of opening their stomata at night. When it's cooler out and more humid, they can take in CO2 without losing too much water, and like the C4 plants, they will fix that, make it organic, store those organic molecules, and use them later in the Calvin cycle to fix carbon dioxide and make glucose. So here we have a side-by-side figure showing a cam plant on the right and a C4 plant on the left. In both cases, the carbon dioxide is entering through the stomata. It is being fixed in the cycle prior to the Calvin cycle. An organic acid is made and stored, and then that organic molecule can be broken into CO2 as needed to supply the carbon for the Calvin cycle.